Carnivorous plants have always been an interesting topic of study in biology. They are plants that are able to attract, trap, and digest prey in order to take in certain nutrients. However, even though they all have these features in common, these features of plants did not come from a common ancestor, but instead came from convergent evolution. Because of this, scientists Fukushima, Fang, and Hasabi wanted to study them to determine what the molecular basis for carnivory in plants was. Before we go into the experiment, it will be helpful to really understand convergent evolution. Convergent evolution occurs when organisms evolve the same traits separately of each other. In other words, it occurs when organisms develop the same traits without sharing a common ancestor that had those traits. In this experiment, the carnivorous plants studied developed carnivorous traits from convergent evolution. The experimenters used Cephalotus follicularis as their model organism. The cephalotus is a carnivorous plant native to southwest Australia that is very useful for understanding carnivorous plants because it creates leaves that are used for carnivorous activity and normal plant leaves. To start understanding the model organism, the experimenters first sequenced the genome of it and compared it to the coffee genome, a non-carnivorous plant whose genome had already been sequenced, to see the differences between the two. They found that there was almost an exact match between the two, but the cephalotus was hexaploidy, like other core eudicots, and the coffee genome was diploidy. The researchers then focused on the protein-coding genes in the cephalotus alone. Because the plant can produce both flat non-carnivorous and pitcher carnivorous leaves, the experimenters wanted to know what genetically caused the plant to grow either flat or pitcher leaves. After adjusting the temperature to make both pitcher and flat leaves grow, they compared the mRNAs that were transcribed from genes in both types of leaves. They found that the transcriptomes in the flat leaves were consistent with the gene ontology terms for photosynthesis and that the transcriptomes in the pitcher leaves showed carnivorous features. This proves that in situations where the plant needs more glucose, the plant will grow flat leaves, but when it needs nutrients such as nitrogen, it will grow pitcher leaves. After having discovered the genetic differences between pitcher and flat leaves, the experimenters compared the model organism to 3 other carnivorous plants. They wanted to explore the digestive enzymes secreted by carnivorous plants through a comparison of Cephalotus, Drosera adelae, N. alata, and Sarracenia purpura. These species were specifically chosen as they were generated from 3 separate carnivorous plant origins. To understand this concept, it is essential to understand phylogenetic trees. These structures demonstrate the evolutionary relationships from a common ancestor to a specific species. The type of tree here is a polyphyletic tree because it doesn't include a common ancestor, just several species. Through the sequencing of digestive fluid proteins, the subsequent proteins all developed orthologous ties with the plant carnivores. Thus, despite differing plant ancestry, these genes were continually utilized for carnivorous digestion. The researchers continued to explore these findings, making further comparisons to previously sequenced genes and discovering that the proteins are responsive to environmental stress. Thus, it was inferred that carnivorous proteins came from those stress proteins. The plants using their proteins to respond to external stress is an example of ecophysiology, showing that the environment affects the organism's response. Subsequently, they delved into the details of the digestive fluid proteome and these proteins, finding 3 aspartic proteases from the cephalotus, which carried genes for pitcher leaves that continually are expressed. In order to compare how each of these digestive enzymes function differently amongst the plants, phylogeny reconciliation was utilized. The conclusions drawn from these findings emphasize the manner in which genes are duplicated and how separate ancestries of carnivorous plants share similar digestive fluid proteomes.